And we are fortunate today to have with us Linda Ruben, who's I don't want to say an old time friend of mine, but a person who's been with AFA for a long time and been one of my best friends here to speak on genetics. And Linda, you have to give her so much credit for the fact that she is so well known in the avicultural world based on all of the various and sundry publications that she's been involved in and the many groups that she's headed up. And I would like to remind you that she's done all this on her own. She's a self-learned individual. She doesn't have a PhD, although I'm sure many PhDs would be envious of her knowledge in the area of genetics. So without further ado, I'd like to introduce to you my friend and your speaker tonight, Linda Ruben. Linda.
Thank you. And thank you, Tom Marshall, our former past president of the AFA. I appreciate the introduction. Today we're going to be looking at a new nomenclature affecting the color mutations in cockatiels. And there's quite a bit that's happened. So I'm going to try and present this in a way where you'll see as we go along. We'll start off with some of the main mutations real quick. Then we're going to hang on to your seats because we're going to see a lot of stuff. Okay, just to begin, we need to know about pigments. Cockatiels operate by two major pigments. The first is melanin, and it's responsible for the black, brown, silver, tan type colors that you come across. The second group are called citizens. Now, you may have heard them described before. Carotenoids, that was the form. Dr. George Smith coined the term citizens because carotenoid implies color feeding, which cockatiels are not color fed. And carotenoids are something that feed birds in the forms of carrot or shrimp or whatever. Cockatiels do not have any influence from food groups. Citizens was termed because it applies to parrots. So those are responsible for the red, orange, and yellow in the birds. Now, when you don't see any of those colors and it's just white, that shows you that it's the absent of all pigment. Okay, real quick, normal gray is the nominant cockatiel, the dominant form, and please do not refer to it nominate. We don't nominate the cockatiel. It is a nominant, as all wild forms are. Now, there are some standard color mutations you're all familiar with, and they're listed there. We'll take a quick peek. But first, I just want to mention that at least in the nominant form, our birds are sexually dimorphic. So you do have birds, like the male has the yellow facial mask upon maturity. Female lacks it. The male will also lose his under tail markings and his under wing flight spottings. And those can be either yellow or white, depending on the amount of citizen pigment in him. And the hen retains it. Okay, in the pied mutation, it is actually a color mutation. It's a leucistic form. We don't really refer to them as a pattern, but they do range in the pattern or the appearance of the yellow or white wash that's on these birds. I tried to find light pied for you. I couldn't really find because a lot of people are breeding heavier pieds. These are my guys, and I haven't bred a light pied in a long time. But when you go for a medium to a heavy pied, the idea is you want symmetry of markings, a good balance, one side's the mirror image of the other. And there's a lot of talk about saddleback pieds. There should be a little bit more gray there to make it perfect. But that's the pattern that seems to prevail in a lot of tastes. Now, in the Lutino, we start getting into some of the jargon you're going to hear in the nomenclature. Lutino is actually on the locus for the alleles for Eno. So whether it's albino or Lutino in the bird fancy, that's the locus site that we refer to. So it is an Eno. But these star lutino. They have the red eyes. They do darken upon maturity. A little bit of melanin pigment comes in later and colors them up. And basically, you have a combination of yellow and white. Some people breed more for the yellow. You're going to see that in a minute. And the real, challenge with these birds and one other variety is breeding out the bald spot. So that's something you can accomplish in 3 generations if you selectively breed correctly. Now, the cinnamon is what we call a melanin-colored bird. It's modified pigment. Even the eyes at birth are plum, and that's how you can distinguish them in the nest. I choose to go with some scientists who believe that this is pheomelanin pigment. I am going to be referring to Dr. Terry Martin's book quite a bit and going into some depth on that. He does not believe in the pheomelanin pigment, but I can say that in Dr. Petrak's book, which was the Bible written years ago, in the chapter of her contributing author on genetics, we found out that there are over 300 compounds to yet be named. And I think that for now, pheomelanin is probably acceptable, but we may learn even more. So nothing is in concrete. We're going by what we know now, and that is very little. Now, in the pearl, or as it's known in some parts as Europe, they're trying to, Dr. Martin is trying to bring up some of these standard forms with new language. It's going to be up to USA Aviculture whether to accept it. But it's been known for years that they were called opaline in Europe. And they're trying to keep it that it's the opaline gene that's affected this bird. Now, this is so far the only pattern that you know in cockatiels. This is a pattern bird. And as you know, they have yellow, white, yellow, or white lacings, which is golden pearl, silver pearl, or a combination of both. And they're extending down from the nape down the back, across the rump area in extensively marked birds. And in heavy birds, we call this thrush marking when it spills over onto the front. The outline of the feathers are in gray, the inside is white or yellow. To me, and this may or may not be found the case, but to me, this reminds me more of a spangle budgie than it does an opaline budgie. In an opaline budgie, you'd have a clear mantle. The color of the bird would be in the wing area, but of course, we're working with gray birds anyway. So, but there are lots of different points. I won't get into a lot on it, but I do wonder whether we should be calling this spangle. Now, later you'll see a spangle, but that bird will have different types of markings. Okay. Now, one of the earlier, or I should say, second mutation to follow the pied mutation, which was the first, was the recessive silver. And this bird, as you can see, is being renamed, at least by Dr. Martin, as ashen fallow. The reason he does that, and it's important for us to understand, and I'm trying to share this with everyone, is that all fallow means, and I know this from my work with budgies, and please forgive me for adding that I started in budgies, and that's where I learned everything I know. And so working with cockatiels after that was really going steps back. Budgies have everything. But we have German and English fallows, and in the fallow, in budgies and all other citizens, you have the red eye, and you also have a reduction in the melanin pigment. And that is what classifies it as a fallow. So in the recessive silver, you have both those things going on. So I can understand Dr. Martin's attempt at reclassifying this as a fallow. Ashen is taken from the word ash, soot, a light gray. So that's why you have ashen fallow. The feet are depigmented, the beak is light, and the only other thing I'll mention as I write in my books, I keep bringing up the fact that in Europe, when they came out in the 60s, these birds had a lethal factor and they were born blind. So when the Americans got them, whether it was this dependent recessive silver, we'll never know, but we never had a problem with blindness, so that was very fortunate. Okay, here is the U.S. American fallow. It's germane only to us, the USA, and you have an intensity of yellow citizen pigment and the red eye with a reduction in the melanin. So it's sort of like a cafe au lait coffee type, light coffee type. The eye is red, feet and beak are depigmented. And again, you have that one problem with the bald spot. So you really have to be careful about breeding that out if you're working with fallow. Now, they are being reclassified as bronze fallow, no doubt for the yellow pigment that we see. And here's just some examples of a fallow with a fallow pearl, and you can see the pattern lightly on the back. And I thought you'd like to see fallow or white-faced bronze fallow, as we'll now call them. Now, the whiteface, as you know, is a bird that is void of all citizen pigment, so there's no orange cheek patch or yellow facial mask on the male. And I just wanted to point out in a close-up of the female, see the white cheek patch and a little bit of white on the face? Well, every single book you're going to read is going to tell you there's no white. Just like in the normal counterpart, there's no yellow on the face. That's not true. They do have that insipid marking of a white cheek patch, and I don't know why people don't document it. It should be documented. And you'll see little white flecks, and that can get confusing if markings will be on the back of the neck. But that is a separate issue where a bird is heterozygous to another color. Okay, now we have the white-faced bronze fallow, and it's just another example of an American fallow that has the white-faced gene affecting it. So now it retains the red eyes, the lessened pigment of melanin is reduced, and then you also have the elimination of the citizen yellow, which the fallow is famous for. Okay, after the whiteface came out in the late 70s, for those of you who weren't here, I remember when they did, and in fact, one of the first few people who got it, and I know Tom will remember, it's Nancy Reed. She imported some in too and got them from Dale Thompson, who was the original person who imported the whiteface directly from Europe, and they came right out of the Netherlands. And in fact, in 1982, which was my first talk at the AFA, I had the original slide that Dale Thompson gave me of the whiteface and the whiteface Lutino or albino. And that was such a thrill back then. The next thing to really shake up Cockatiel was the dominant silver. And I was fortunate because I got a lot of information on some of these mutations coming out because at the time I was writing a column for Cajun aviary birds in the UK. And so people would read my column there, would write to me, letters got passed on to me. These were the days of handwritten letters. And I know you don't know what those are. But anyway, so, you know, I got a lot of information about them, and they turned out to be codominant mutations. And when I say that, anything that you see here that says autosomal merely means it's not sex-linked. That's all you have to remember. If it's sex-linked, it'll say so. So on the autosomal codominant, you get two forms of birds, two phenotypes, even though they're genetically dominant silvers. And what they're trying to do is rename this in Australia and Europe as the dominant edged. And that refers to this type of deposit along the ends of the flights in particular and through the body, but primarily expressed down on the secondary and flight feathers, where you have that heavy edging. So it does look like there are additional melanin deposits, or at least they show up because of the reduction in melanin through the body. So the other thing to point out on this is that the bird, oh, I think we went forward. Let's see if we get back. Oh, this is that one. No, okay. Well, we'll skip through. But the dominant silver also has the deposits up on the head, so you will see the skull cap in other pictures. All right, I wanted to mention some of the scarcer mutations. It doesn't mean they're impossible to define. It just means you're going to get them through breeders rather than find them anywhere else. The dominant mutations are a new mode of inheritance for cockatiel people. So we've got to get used to that. And I'm going to comment more on that as we see some of the other mutations. But keep in mind, there's more than one type of dominance. The cheek patch mutations came along, and we got a couple of them, yellow cheeks and different modes of inheritance, pastel face and gold cheeks, which have caused some excitement in the fancy, and they're being bred in various forms. And I call this the silver solution. There are now, if you want to count everything in the world, 8 forms of what we call silver, some of which may be actual silvers and some of which may not be. And we'll address that. There's the new US mutation that I have written about for some time, and I had worked with the birds, which I will get to. A little bit about melanistic birds and a little bit about the citizen spillage. I'm so used to saying carotenoid spillage, and I have to update myself on this, as we all must do. And look at that. Okay, yellow cheek. There's a modification in the cheek patch color only in the yellow cheek. The first ones came from Germany, and they are sex-linked. Their cheek is described as a bright yellow. It varies. It depends on the stock. And let me preface this by saying that many of us are working with the cheek patch varieties, and we're interbreeding them when we shouldn't be. The idea is to keep each cheek patch variety, including through its mode of inheritance, separate. By breeding them together, we're getting this mishmash combination where we're no longer going to be able to identify which is which. So it's important to keep them separate. It's fine to breed them into other mutations. This happens to be yellow cheek pearls, and that's fine. Just don't breed a yellow cheek to a pastel face. Okay, here we have birds from Thailand, and they're being called yellow face. And one on the left is a cinnamon pie. The one on the right is clear pie. But you can see it's really a yellow cheek, if you want to be genetically correct. And the amount of yellow citizen pigment through the bird is controlled through selective breeding. But nonetheless, altogether, it forms a bird to date that's the closest all yellow bird that we've got. So some exciting things can happen. Now, the next form of cheek patch variety is the codominant yellow cheek, and supposedly that's USA bred from Florida. And I remember when there was a lot of exchange in the National Cockatiel Society Journal about whether or not it was codominant, and people were writing back and forth. Supposedly, there is more orange in one form of them. Codominance produces 2 phenotypes, 2 different visual looks. And you'll see that in another mutation in a minute. But what you want to do is make sure that you're working with a form that does have 2 different phenotypes. Otherwise, it's not codominant. Okay, the codominant yellow cheek is being called tangerine. Now, normally I would really protest a name like that, but I have been led to understand that tangerine is an accepted name across the board in other citizen colors. And although it doesn't sound very scientific to me, it at least is one name that people are agreeing on. I tend to doubt that we're going to start using some of these terms like that because the ones we use have probably, but at least to be aware of them and know what we're dealing with, because you may see an ad elsewhere in the world from Europe or the South Pacific or whatever, and they may say that. And again, the question is, are they truly double factor birds? Now, the pastel face came along, and that's a bird with another altered cheek patch. I was in correspondence with Terry Cole and others who had written to me, and they described these things as they were breeding them, and they said that it was more like a peach color to the cheek patch. Again, the birds we see today are very hard to see that defined color as described. I leave that to you, but you have to keep them separate as much as possible. Now, it's interesting when you work with pastel face, also known as pale face genetically, that it's on the parblue locus. So it does interact with white face. And this is where we get a lot of confusion because people say that pastel face is dominant over white face, and that is not true. Both pastel face and white face are autosomal recessive mutations. It happens that when they interact, there's a hierarchy of recessives. Think of the white face as a double recessive. That's the best way to think of it. The pastel face behaves in a dominant manner over the white face. And so you can have a pastel face split white of a white pastel face. Okay, another parblue allele of the blue is a patch mutation, and it's hard to see here, but it's called the gold cheek. And that's relatively new. Barbara Green out of Virginia was working with the birds to establish them. And again, not a lot of people have them, so we don't have a lot of information. This was one of mine. I didn't get a chance to breed her. She came down with diabetes, and I lost her. So, it's very difficult at this point to come up with anything that is that factual about them yet. Okay, the pastel face is pictured here against the gold cheeks. You can just see a little bit of difference in the cheek patches, but again, it's not a real exciting, visual when you look at parrots, but for cockatiel people, it adds a little interest. Okay, again, it's time for the silver solution. And we have birds that are referred to as recessive silver, now called ashen fallow, plus 6 more. Okay, here we come to what I call the suffused silver, aka emerald. Excuse me, I'm jumping ahead. Very sensitive. Let's see if we can get back. Yeah. Okay, we have melanin altering genes at work again. And there are several points about these birds. Again, nothing is set in stone yet because there are some people arguing as to what they are. And I got to cut this out. Let me put this down. Take it that way. Okay. If you notice, the birds are lighter on the back, dark on the rump, darker on the front. Males always lighten with age. The backs get lighter and lighter. This is the male here. Basically, you have a bird that has a reduction in melanin. Whether or not it's a true dilute is, being under discussion. I think of them as a dilute. I think genetically they're a true dilute gray. I know Dr. Martin would argue that with me, but we have different birds. And frankly, when you work with a bird for years and you're breeding them against someone who's only looking at pictures of them, I think you get a little bit more information. So, um, again, they have several names in Australia already that they're sticking to, and they have their dilute gray. So they may go on and keep that. But I think for our purposes, we may never get those colors, and we have certain classifications and categories, and I think that's what we're going to end up doing. This is the white face form, and you can see, in the combination that you have a little bit of a skull cap and edging, depending on pictures. This one doesn't show it as much, but the bird at home does indeed have a lot. And then you can see when they feather out as babies, you get a different look at, there's no um, citizen pigment because of the white face, but you also see some of the edging in the feathers. Okay, here's an interesting one. I don't know if anyone's ever seen these out anywhere, but there are a couple of breeders working with them. And they're called black-headeds. And what they are basically is a combination of the dominant silver with what you know as emerald. Again, emerald is not a scientific term. It was a name the breeder gave it because of her belief system. And basically, there is no emerald in any other citizen bird. It just happens to be a name she likes. But I will say, Margie Mason established the variety. It was given to her to work with, and she bred them in just about every form. And if you check back in the NCS journals when I was writing as genetics consultant, in the beginning, I refused to classify them, and I said we need a lot more work with them. They need to be bred with fallows for the simple reason of finding out whether fallow is affecting their phenotype. And I still don't know the answer to that question. But we were under publication deadline, and I had to come up with a name, or else. So I gave the name suffused yellow, simply because I could see a lot of the yellow. I worked with suffusions and budgerigars, and I've noted that in some of the white series, like the sky blue, cobalt molds, and even violets, there are light, medium, and dark suffusions. So I will not accept the fact that, oh, there's too much melanin to call it that or whatever, because there are different levels. In the black-headed, you're combining that emerald or edged, let's see back, here we go, with the suffused, with the dominant silver. And so because of that, that skull cap comes through for some reason even heavier. The deposit of melanin somehow is more intensified, and there's a further reduction through the body. So you look like you have a black-headed bird, and there are some pictures, I haven't included all of them, but there are some that look quite dark, so don't be surprised when you come across them. Okay, now supposedly we don't have these, but that's a bird from my aviary, and in Australia they call it a silver spangle. It's believed to be bred out of the suffused silvers. At least they got mixed in somehow, and that's, I believe, is how I got one. Um, anyways, they're autosomal recessive, and they're called edge dilute because the markings are primarily along the flights in the back, and you can see those deposits of melanin that give that edging. And that's basically the mutation, the darker edged feathers. And just for comparison, that's a white face variety of the edge. And again, we would probably refer to them as the silver spangle. That's what we're familiar with. Okay, and here we have an interesting combination. This is in an aviary from someone who's worked with these varieties for probably about 15 years. It's a white face, single factor dominant silver, dilute, edged, or silver spangled. I know that's a mouthful, but it's 3 mutations. So white face, dominant silver, edged, or silver spangled, all 3. And here you have a nice wing shot of the silver spangle in the pie variety. And it shows that edging, that deposit. And we go on to another silver variety, and these we don't have. These are in Australia. When I was out there speaking in '97, which is ancient history now, I did see a lot of Australian platinums, and I saw them primarily in Australian platinum pearl. Very few just straight platinums. Now, that gets confusing because in the UK, they were breeding the dominant silvers to their white face. And that combination they would refer to as platinum. So you have to be careful what terms you're using, whoever you're writing to or dealing with birds. They are sex-linked perinos, so that means that basically they're on the locus. And because of that, when you breed them with perinos, you'd be donating one Australian platinum gene and one Eno gene, and they act codominantly together just in behavior. They're still, of different modes of inheritance. And you get one of each type thing. And that was the big dilemma. Why is it we breed this to a Lutino and we get one of each? Well, they're one's sex-linked and one's, a sex-linked. And it shouldn't work that way. But that's how they do turn out. And again, in the Australian platinum itself, what you're doing is you're getting a reduction in the melanin pigment to about 50%. And so it comes out to be like a smoky gray color. And this is the Australian platinum pearls, so gives you an idea. Very pretty birds. And one day maybe someone will bring them over here, or we'll luck out and they'll, if they the ban, which is never. And but there are, breeding consortiums, who knows. And the other thing would be that maybe a spontaneous mutation would happen here, because again, a lot of the colors we have arisen spontaneously in Australia, and they can be very, very similar, although not all of them are 100% like ours. They have their own phthalates. They have their own types of silvers, which you'll see further here. So this is the combination with the Lutino. They call them Plutinos for short, and you have the bird right here. It's sort of not in the best light, but it was a friend who sent me their child's bird. It's not in the best condition. But you can see how reduced the pigment is there. It almost looks like a Lutino. Okay, and I just wanted to point out the difference. When I was over there, they kept asking me, we heard about your U.S. mutation, and we've been told it's the same as our mutation, the Australian platinum. So what gives? Well, if you look at the birds just very surfacely, you have Australian platinums have a darker back. Our mutation has a lighter back. The Australian mutation has a lighter rump. We have the darker rump. On the undersides, the Australian mutation is lighter, ours is darker. So it's exactly the exact opposite of what we have. So once they saw the birds I showed them, they were, oh, okay, we get it. Now that clears it up. We're talking 2 different mutations. And there had been people here in the U.S. that just looked at pictures and by pictures said, oh, yes, it's the same 2 mutations. So it shows you how dangerous it is to rely simply on photographs. It doesn't tell the whole story. This is what they call the olive suffused in Australia. Reduction in melanin. Dr. Martin feels it's say dilute. I think it's too dark for that. However, again, you see the pigment coming through there. It would be because of the reduction of the melanin as opposed to intensity of the citizen pigment, but it's interesting in itself. Okay, now there are a couple of silvers that we don't have, and they call them the East Coast silver, which is their true dilute. It's a pastel. I don't know if there's been any quantification of pastel, but years ago they hired a scientist, the biggest European avicultural organization had 50,000 members. Can you imagine that, Tom, huh? And we should be so lucky. And but that was back in the 60s, and they hired a scientist, and he quantified the melanocytes in the feathers to find out what degree or percentage qualified for dilution versus pastels. And there are specific measurements. So again, we use these terms, but whether or not these feathers are actually taken and examined and quantified that way would be another story. And some people claim that they have enough information to suffice. Excuse me. But again, this would be the East Coast would be a true dilute. Now I saw the West Coast silvers, and to me they simply look like our recessive silver with black eyes. That was the only difference. But the interesting thing about them was that these birds were not a spontaneous mutation. They were selectively bred, and people paired their lightest normal grades to one another until they came out with this bird, the West Coast silver, and the genetic term for that is faded. The idea is that if you kept on breeding them, although they do reproduce as is, but if you kept working with that, you might get to an albino, eno. So because you would reduce the melanin pigment so much. Some of these birds, that are melanin reduced have some involvement with the eno locus, so that they may be alleles of Lutino genes, and this is what a lot of talk is about right now. Not just these, but others that I've been showing you. Okay, again, another possible dilute. It's called suffused silver gray elsewhere in the world, similar names. They have melanin reduction, and that's why you're seeing that citizen pigment shining through. Interesting bird. Now, another one that came to light a couple of years ago here was the cream face. That's basically a bird that has a bit of yellow, white on the face, not enough to be a yellow face, or it's not affecting as much the area. So in that respect, we call it a cream face, and in the Lutino series combined, we call it a creamino. Okay, now this is a little bit exciting. This is something I almost wasn't going to show you and tell you the names and go, does anyone know what this is? But I won't do that. Basically, the question is it a chamel? Because the scamel is a bird that is a combination, a merging of several colors together. It's rare, it's unusual, you don't breed a lot of them, you're lucky to ever get one. But they exist in other parrots, and I've seen them more than one in cockatiels, not at shows, but people bringing pictures to me and say, what the heck is this? So that would be my question, is it a true female? Because those are supposed to be defined as mottled with a blending of colors. So that would be the definition coming out of a term from Germany. The bird on the right may or may not be, but it looks like it might be in another form. I wasn't sure if that eye might even be plum. It's hard to tell sometimes in photos. No, hybrid has nothing to do with it. Hybrid is two different species. A mutation is within the species. It's still a mutation. And the citizen spillage is not a mutation per se. It's just an overabundance of the citizen pigment. And that can happen to birds that are breeding around this area. But I included to show you that sometimes this village can go deep. And I have heard of orange birds down in Africa, so nearly completely orange. So that would mean that people would selectively breed for that. And whether or not we could always do that with just a tiny bit of spillage and work our way to bigger things, I don't know. But we'll find out in time. Or whether these orange birds that we hear of that are more deeply orange than this are an actual other mutation, we'll find out. And I included this to show you. This is what I call a tricolor. Again, you have a bird that's a normal pied, or is it a cinnamon pied, or is it both? You have white, brown, and black. See the brown on both sides. I was lucky enough that when I judged a show in Puerto Rico, I didn't have just one. I had a team of these tricolored pieds to judge. And I'll tell you, it was pretty amazing. But it shouldn't happen. I wondered, is it a half-sider gone wild? Is it something of that nature? Because a half-sider, and I don't have an actual picture here, I do in other parrots, but the half-sider is actually a genetic mistake. And it's something where you'll see birds, say ringnecks or budgies, they'll be green on one side, and as if a vertical line goes down the center, and the other side will be blue. And what it is basically a mistake in sex cell division, or during the time that the zygote is forming, and the bird really should be blue or it should be green, but a mistake takes place, and so it really should be green split blue, but the blue shows. And so when you breed that bird, you're not going to be breeding replicas of it. It just won't happen. It's an anomaly. But something like this, we don't know because half-siders are divided by half. Quarter-siders are usually the upper quadrant of the body. So, and that's all we've ever seen in a variety of parrot species. So I have to wonder what this is and what's going on to permit two melanins to show up. Is it the eumelanin in that variety, because it is a dark pigment, or is it in combination with pheomelanin or another pigment we have yet to classify? So those are some of the questions I come up with. And just real quick, I want to remind people that working with stock, always get the best stock you can afford, even if it's just 2 pair, rather than buy 20 birds that are not good quality. And acquire enough birds that you can start a line with to offset any losses, because there can be losses down the road. And if you put everything by splits, invest in splits, but know your source and get references, because people will tell you anything sometimes to sell a bird. So find out who's reputable and reliable and stands by their reputation. Uh, you want to measure your progress in working with any mutation, so pair compatible birds, and that's not just will they breed, but also what the right colors are. You don't want to start breeding all those cheap patch types together. Assess your birds side by side, just like you would before a bird show. When you do your breeding pairing, put them in separate cages and figure out their good points and weak points. Write down your objectives, or else you'll never remember them. That bird looks good with that bird is not a good breeding program. And keep your stock for at least a year or two before you part with them, because they're not going to show all their qualities for some time. Cockatiels are parrots. They're not going to flesh out in 1 year. It's going to take them 2 to 3 years to fully show their entire development. And we have a saying in the fancy, keep the best, cull the rest. And it's very true no matter what species you're looking at. And above all, keep meticulous records, because take it from someone who used to pride themselves on their memory, you don't remember things after a while, and you'll forget the details. So record keeping is a must. And please, please, please give pedigrees when you sell your birds. A lot of people are stopping that. They're getting lazy. So make sure you give a couple of generations as to where your birds are coming from. If you want to know anything else about the mutations, I do have a digital publication that's available on my website. I'll just mention, so you can always go there. And I just wanted to acknowledge the many people who contributed to the program with slides. I appreciate that very much. And it really adds to a program when you can include it along with your own. So thank you very much. I enjoyed speaking with you today. Sure.
One question I have would be, since you had the advantage of being in Australia, did you have the Australians tell you whether the normal cockatiel was being affected by the mutations, or are they been able to retain their same vigor and their same physical size?
In the wild or in
When
captivity?
they have the ability to look at the ones in the wild, and they have the
Right.
ones that look at the ones they've been dealing with. What's happened there with that?
Well, one thing, and I should have thrown in a few slides on it, I forgot, different programs, but it's very exciting. When I was there, we went into the outback of St. George, and I saw the cockatiels in the wild, and it was a lot of fun because we actually followed them before we went into the paddocks. There was a flock that was in the rural outskirts, and we were able to follow them to a grain mill where they found some sacks of spilled seed and they were feeding. So we asked the people at the mill, could we drive them back and get a close look? They said, sure. So we went over and we looked at them, and the birds were, they amazed us. We got pretty close. We used binoculars and we had a photo lens as well. And those birds were as sizable as any good quality normal was. There weren't these little birds that you see in pet stores or whatever, in the poorer ones. These were sizable birds. And amazingly, you know what the biggest normal was? Tick mark, split pie. So that kind of answered a question for me about mutations in the wild and the survival of the fittest, and is big too big? So a lot of these questions get answered for you when you actually see the birds out there. In captivity, the normals, to be honest, I didn't see a lot of normals. I saw a lot of silvers, a lot of Australian platinums, platinum pearls, West Coast silvers, oodles and oodles. I had the fortune to stay with Mike Anderson, my host, for the first week in Brisbane, and he had an entire backyard acreage of a supermarket areas. And we went through looking through the boxes, the babies, and primarily silvers and, yeah, a few other mutations, but that's what's popular there. And those platinum pearls were incredible and, made you drool and say, why can't we have them? But maybe one day. And then I went on to Sydney and I stayed a week with my host there. That's where we had the International Symposium, and I was lucky to be keynote there. That's why they brought me over. And so I had the joy of getting to speak with my co-speakers, which were Diana Anderson and Peggy Cross of the first edition of the book you might be familiar with of Australian Mutations in Pet and Aviary Birds, which Dr. Martin did the second edition. And I'll remark, he did a heck of a lot of work. I may not agree with him on everything, especially our U.S. mutation, but I credit him with doing an awful lot of scientific work, which was wonderful. And it's a good read. But I got a chance to talk with the original authors, and I will say I had some fascinating conversations, unlike any I've had in America. So it was a lot of fun. And then after that, I got to go on to Melbourne for a week and speak to the Australian Avicultural Society out there. And I brought in stuff about AFA and legislation, as well as talk about reproducing cockatiels and parrots here and touched on mutations. Because interestingly, in the bigger organization, they were not as keen on mutations because they thought of them as hybrids and they didn't fully get that. That was two different groups, old timers versus, to me, the younger people were with the mutations. So it was an interesting experience.